Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. I was discussing about the citric acid fermentation process. Also, I discussed about the submerged fermentation process, which is largely used by the industry. And then what is the that flow diagram of this whole process, a process flow diagram that I try to discuss both process flow and block flow diagram. And we have come across that we have three part we have in any industry. We have upstream processing, then we have bioprocess, then we have downstream processing. Upstream processing mostly deal with for the preparation of the media, air sterilization, medium sterilization, and inoculum preparation. And downstream processing usually deals with the purification of the products. Now, in case of citric acid industry, there are so many downstream processing involved. Now, we'll discuss those processes in details. Let me first discuss that how the calcium citrate precipitation take place. This is citric acid, and this is calcium hydroxide. Lime, when in combination with this, it produces the calcium citrate, and then this calcium citrate, when we hydrolyze with the H2SO4, concentrated H2SO4, it produces again citric acid and calcium sulfate, 2H2. This is called gypsum. And calcium hydroxide is usually produced from calcium carbonate. Calcium carbonate, if you heat it, it produces calcium oxide and carbon dioxide, and then gives calcium oxide. When we make a slurry in water and filter it out, then we produce the calcium hydroxide. So this is the kind of stoichiometry of this process. This will be very much required to know that how much calcium hydroxide is required for the precipitation of lime. We can also find out how much of sulfuric acid required. We can also calculate to find out how much gypsum is produced in the fermentation process. Now, another sample calculation I want to show you in the particular citric acid fermentation process. Suppose we are using 200 cubic meter of this batch fermentation process, and sugar concentration is 200 grams per liter. 200 grams per liter, that means I showed you before that if I write the initial substrate concentration is 200 gram per liter, you can always convert it to kg per cubic meter. How we can do that? I can write 1 cubic meter is equal to 1000 liter, and 1 kg is equal to 10 to the power 3 gram. So this gram will cancel, this liter will cancel, this will cancel. So this will be equal to 200 kg per cubic meter. So you can easily convert to that unit what we have done it here. And then sugar input in the fermenter, this is the volume, 200 cubic meter, then 200 you can multiply it, and if you divide it by 1000, then you will get this will be, if you multiply, you will get the kg, but if you divide by 1000, because 1 metric ton, MT is the metric ton, is equal to 1000 kg. So you can, this is the total metric ton of cane molasses required for one batch is about 40 metric tons. Now, in the fermentation process, I forgot to mention that all the fermentation process, we should have a rapid estimation technique. I can give the example of citric acid industry. Actual citric acid estimation process takes 1 day. But when your plant is in operation, It is if you take your decision after one day, it is too much. It is not. You will be having lot of loss in the in process. So we shall have to find out a rapid estimation technique. It is not exact estimation, but it gives you some information whether your citric acid concentration during the fermentation is increasing or it is coming already in the plateau. So for that, what technique we use the titrometric technique. We use we use some sodium hydroxide solution, and we try to titrate that, and we find out that how much sodium hydroxide one normal sodium hydroxide or 0.1 normal sodium hydroxide solution required to increase the pH to 8.6. We have a standard chart. From that we can find out that if this much milliliter is required, this is equivalent to this much of acid. So from that we can easily calculate how much acid is there. Now if you during the and this procedure hardly takes couple of minutes. So this will help us to find out that whether acid formation is increasing in the fermentation process or not. So when you will find that it is almost coming constant, then we stop the operation and do the harvesting. So I want to point out that total acid concentration we usually estimate in the industry. So here we have that I try to find out that how we calculate the citric acid and total acid residue. Now this is the regular sample. This is analyzed by taking the sample at the regular interval, total acid concentration, citric acid concentration. Finally we do that. Let us assume the total acid concentration after 124 hours of fermentation is 14.82%. Now this is weight by volume. That means if you actually it will come 148.2 kg per cubic meter, and that is equal to 0.1482 metric tons per cubic meter. Now if the citric acid concentration is 14.15%, because it will be little bit less than that, then it is coming. This is the typical estimated value in the industry I am showing you. And then this, if we assume the downtime of the fermenter is 12 hours, and then we can easily calculate that what is the yield of total acid. How we can calculate the yield of total acid? Total acid production will be what? This into 200 cubic meter. This is the total acid. Am I right? So total acid, this is multiplied by the an amount of that sugar. How much input is there? 40 metric tons. So we can easily calculate, and we multiply by 100. You can find out how much this yield is there. Percentage yield we can find it out. Then the yield of CAM, that citric acid monohydrate, we can also calculate similarly. We multiply, this is 70.74. So this is how we can calculate total acid yield and CAM yield in the citric acid industry. Now we can also can find out what is the ratio of CA and TA. This you can find out, this is 0.95. The productivity, now how we can calculate the productivity at 124 hours, that also we can calculate. We know this is the total amount of this citric acid monohydrate that is produced, and this is time of fermentation. This is 12 hours is the idle time. If you divide by that, you will get 0.208 metric ton per hour. So this is how you can calculate the productivity of this. Here I want to point out two different things. The actual volume obtained after the emptying the fermentation, which is less than the filled volume due to evaporation. Because since we do the aeration in the fermentation process, there is every possibility, some due to the saturation of the air, some of the water present in the media, that will be evaporated out. The volume will be decreasing. The mold volume also assumed to be 2% of the fermented filled volume, because the mold fungal cells also occupied some kind of volume in the media. That also that is to be corrected. So these are the couple of things. Now let me go the details of this downstream processing, how the cell mass separation, how the calcium citrate separation, all this thing is taking place one by one. Now this is first I want to show you the rotary vacuum filter. the filtrate of the culture broth and washing mycelia, which maybe contains about 10% weight by volume citric acid. So what is happening? We take the filtered the fermentation broth here in the trough. This is the trough, and this is the rotating disc. And now here this is the pipeline, and I told you this is the fine muslin cloth. This is dotted one is a fine muslin cloth, and this pipe we applied vacuum here. So as soon as we vacuum here, the water will be sucked, and since we are sucking water contains cell mass. The cell mass will adhere on the surface of the muslin cloth. Then you can also clean it with the washing water, so that some citric acid which is attached to the cell mass, that also that also we can recover. Then when it come it here, it will be perfectly dry, because since we are applying vacuum, it will almost dry, not completely dry, that almost it will be a lot of moisture will be taken out. Then a knife will touch the muslin cloth here, and this will separate the solid. Now here we have a screw conveyor which can take the material from here to the wagon, because in the below this unit there might be the relay wagon where we can put this cell mass there, and this cell mass is used for the production of raw quality paper. So this is this can be used for the raw quality paper formation. Now then another step is the calcium citrate precipitation. I mentioned that calcium hydroxide is added until the pH is 6.8, and calcium hydroxide neutralizes the broth and forms the insoluble precipitate of calcium citrate. That is, the calcium citrate contains about the 74% of citric acid. The calcium citrate then was heated and filtered. Now how it taking place is like this. This is called Panavis filter. Now let me explain it. This is thick cotton pad. This is the white one is the thick cotton pad, and inside you can see this one. This is stainless steel. This is made of stainless steel, and there we applied the vacuum. Here we have applied the vacuum. When your Panavis here we put the slurry containing calcium citrate. And when you put it here, then what will happen? Here we apply the vacuum. Since we are applied the vacuum, it suck the water, suck the liquid here, and this is the filter, and this is the industrial effluent, and then this is the solid material. This is solid material. This is the calcium citrate. This automatically they fall it down in a then with the help of screw conveyor we can take the material one point to others, because screw conveyor looks like this. This is the shaft, and screw is like this. This is on the surface like this. So this is inside this vessel. So here if it may put it here, and if we put it here, so it is connected with the motor like this. We have motor here. So and here we have also bearing. This is all bearing that we have. So we can have the bearing. So the material will fall here and take the material out. This is used as to transfer the material from one place to other. So it is called screw conveyor. Screw conveyor. This is called screw conveyor. This is largely used by the industry. Then I told you this is treated with sulfuric acid, and this is a kind of start tank reactor, and calcium citrate react with sulfuric acid, form calcium sulfate and citric acid, and this citrate was separated with the help of gypsum filter. Now let me show you the little bit of gypsum filter, how it looks. Gypsum filter is like this. So here we have thick cotton pad here. So here we applied vacuum. So if we put the slurry here, then what will happen? That calcium sulfate will be here. This is the calcium sulfate. 2H2O. Now there is a handle here, we can handle here. This is mold with some kind of bearing here. This is also kind of bearing here. So you can tilt this. So you can tilt this, and below below you might be having the wagon here. So you can this material will fall down automatically the wagon. This is the relay wagon, and take the material out. And this is, I told you, this gypsum is used as a is used in the cement industry. So then then this citric acid contains some kind of color material. This color material is removed with the help of activated charcoal, and then after activated it passes through the evaporator. And I explain how the evaporator, it is tubular evaporator. It is like this. So here we have steam. This is the steam for heating purpose, and here you have citric acid. So it is comes here like this. Then ultimately we throw it in a particular kind of vessels like this, or like that we have cyclone separator type of things. So when you throw it out, here vapor will go out, and here concentrated liquid will come out. Concentrated liquid will comes out. So this is the process through which we can have concentrate the liquid, and then this passes through, and this is the process through which we can remove the color. We can pass the crude extract here, and this is the activated charcoal. Then we can regenerate the activated charcoal here, and this is again we take it here and fill it, and we get the decolorized extract. This is for now. And then we pass it through the crystallizer. I showed you the crystallizer. That is I told you that crystallizer is simple, the kind of vessel, that reacting vessel, and this is usually the insulated. I told you this is insulated because we maintain the temperature as around 10 degrees centigrade. And I told you the citric acid has a transition temperature of 36.6 degrees centigrade. Lower than 36.6 degrees centigrade, citric acid monohydrate formation will be there. Now this is the agitator, and anchor agitator might be used because which has less friction, because this is usually move very low RPM, 1 RPM. 1 RPM is very low, because if we move this very high RPM, this will affect the crystallization process to a great extent. So little starting is required, just which help in the crystallization process. The degree of purity of the citric acid depends on the purpose for which it is required. For instance, pure form of citric acid needed for the use of food preparation, while the industrial purpose we require crude, because even if its color is there for industrial purpose, now crystals of citric acid looks like this, is perfectly white powder. You can see how it looks. Now citric acid can be produced in two forms. I already mentioned that monohydrate calcium, the citric acid monohydrate and citric acid anhydrous. These forms are required additional purification step to reach the that desired purity. Now monohydrate contains 1 mole of water molecule every citric acid molecule, and requires repeated crystallization under the water content is approximately 7.85%. Now after the crystallization process, it passes through the centrifugation machine. Just in the continuous centrifugation machine, when you pass through the crystals will throw to the periphery of the crystallizer, and liquid will be collected from the middle, and that liquid contains about 30% of citric acid, that is called the mother liquor. And solid crystals, we take it out, we wash it, and then these crystals we passes through the dryer, what you call drum dryer. Drum dryer also looks like this. I can explain a little bit how the this is called drum dryer is used. this dryer is like this. So we have some kind of arrestor like this. Suppose we put the crystals, citric acid crystals here, and here we pass the dry air, and this is rotated at the very low RPM. This is rotated at the very low RPM. That is this is the gear that we have. Then this is geared. This is connected with some. This is might be motor with connected with motor. It is connected here, and this is the supporting material. This is kind of supporting material we have. So it is rotated very low RPM. Suppose it is rotating like this, and when you pass the crystals, the air will go out. Air is out. Air out. So when it rotates like this, and material roll in the drum like this, and air comes like this, and in this way the air that crystal will be totally dry. Then it put it to the sieving machine. I showed you before also. In the sieving machine we have different sieving plates. We have, I told you, maybe the 10 mesh size, they made 20 mesh size, 30 mesh size, 40 mesh size, 50 mesh size. So we can collect the different size of crystal, 10 to 20, 20 to 30, 30 to 40, 40 to 50, and one mesh is equal to approximately equal to 1 by that one mesh is the 1 inch. That is the 1 inch. That means 10 mesh equals to 1 by 10 inch. This is the inch. So this is the port size, because the port size of the mesh. This is the port size. This port size is 1 by approximately, not exactly, approximately equal to 1 by 10 inch. So now question comes how we produce the anhydrous citric acid. The anhydrous citric acid also very interesting. That we take in a this is the vessel, in the reactor vessel. Then we have a starter here, and this is the motor, and we put it. This is the mother liquor. And we put here, we put a cam, and when we put the water molecule present in the cam, that will go in the mother liquor. We will get here, we get the citric acid anhydrous. Now here the temperature we maintain about 40 degrees centigrade, because transition temperature I told you that 36.6 degrees centigrade for calcium. This is for the production of citric acid anhydrous. So citric acid anhydrous production, let me repeat it again. What is saying in this is just a kind of a reactor vessel, that here we take the mother liquor. Mother liquor contains 30% of citric acid, and then we take cam that is crystals we take, we put it here, we put the starting, then water molecule present in the cam, that will in the go in the soluble form, and with the whatever solid that will remain there, that is citric acid anhydrous, and it will just look like perfect powder. It is not like crystals will give you, citric acid monohydrate will give you some gloss, but this citric acid anhydrous is just like a powder. This is used in the pharmaceutical industries. Now I want to solve you two problems which is related with this citric acid industry. I hope this will be useful for you. Citric acid is produced from cane molasses containing 48%. May you consider weight by weight this maybe sucrose, or volume, whatever you consider, because nothing is mentioned, by Aspergillus niger using submerged fermentation process. The capacity of the fermenter is 100 cubic meter. Average time of fermentation and citric acid yield is 148 hours and for 80% respectively. Compute the following. Amount of cane molasses required for the fermentation process, assuming initial sugar concentration 225 gram per liter, productivity of the fermenter assuming the idle time of 12 hours and percentage utilization of sugar. This is to be calculated. Let us see, it is very simple. The capacity of the fermenter is 100 cubic meter, then concentration of the concentration of sugar in the cane molasses is 48%, and then this is the average time of fermentation is 148 hours. This is given. Now idle time is 12 hours and citric acid yield is 80%. So initial sugar concentration is 225 gram per liter. This is equal to 255 to 225 kg per cubic meter. I showed you the calculation before, and then you can find out how much metric ton of cane molasses, because you see that how much this is the amount of sugar required, you multiplied by the volume, and this contains about 40% of this sugar. So it will be divided by 0.48, and then it will multiply by 100. Then this is coming around this 47 metric ton per 100 cubic meter. Total amount of molasses required then equal about 47 metric tons. Yields, how you can calculate? This is the total amount of sugar, that is the concentration, initial concentration. Then this is the volume of the fermenter. If you multiply it, this is this much kg of sugar. How much is the yield? Is 80%, 0.8. So 80 divided by 100. So it is coming about 1000 kg. Now productivity, how you can calculate? This is divided by total time of fermentation is coming about 62.5 kg per hour. So this is how we can calculate the productivity. Now most important thing is that how you can calculate the percentage utilization of sugar. This is the stoichiometry we have. This is the sugar with molecular weight is 342, and this produces 2 moles of glucose, and then it produced 2 moles of this citric acid, and we consider the anhydrous. Then if it is anhydrous, then this gram and this much of the citric acid is produced from the 342 kg of sugar. So we have already calculated before that how much is the sugar is produced, about the total is 1000 the kg. So here we calculated in different way, that we calculated that 0.8%, the 80% that is recovery of this citric acid. So 225 is the sugar. If you multiply by 0.5, then we get this much of sugar is converted to citric acid. Then from this stoichiometry we can find out how much actual sugar is required for getting this much of citric acid. The initial concentration or initial amount of sugar is like this, and the amount of sugar that is required for producing this much of citric acid is this much. If you take the ratio, it is 70.3%. So that is the percentage conversion. Because why I want to show you that this is not equivalent to the actually the yield that we have. Yield is 80%, but actual sugar utilization, when you calculate, we find it is 70.3%. Now another problem also I have solved here, that is 100 cubic meter citric acid fermentation broth has been harvested in the harvesting tank. Broth contains 11% weight by volume cell mass, 12% weight by volume of citric acid. Compute the followings: total amount of mycelium produced, lime required for calcium citrate precipitation, maximum amount of gypsum produced, amount of water to be removed to increase the citric acid concentration 22% to 60%. It is very simple, that volume is 100 cubic meter, citric cell mass concentration 11, that means it is coming about 110 kg per cubic meter, citric acid 120 kg per cubic meter. So you can easily calculate how much cell mass is produced. It is produced around 11 metric ton, is quite high. And then we have the stoichiometry, this much of citric acid monohydrate required this much of calcium hydroxide to produce how much of calcium citrate. So amount of citric acid required produces this much of 12 metric ton of citric acid. Then from this stoichiometry we get for 420 citric acid required 222 gram of calcium hydroxide. So for this much 12 metric ton how much is required, we can calcium hydroxide required we can easily calculate. So this is about 6.34 metric ton of calcium hydroxide required. I told you calcium hydroxide is produced from calcium oxide. Now how much calcium oxide required, that we can just see this unitary method through which we can find out that how much calcium oxide is required. Now finally we shall have to find out how much gypsum is produced. Now this is the stoichiometry we have. This is the citric acid monohydrate with calcium hydroxide produce the calcium citrate. Now question come, how much calcium citrate is produced? It is 16.2. Now this calcium citrate again converted to 516 gram of gypsum. From the stoichiometry we can find out how much gypsum is produced, that is 14.7 metric ton. Then finally we want to find out how much water is to be removed to increase the citric acid concentration from 22% to 60%. This is simple material analysis. 2200 into 16 into V. We find out how much after when you make it 60%, how much volume will be remaining. This much volume will 36.66 milliliter volume will remaining. So how much water is to be removed means 10 minus 36.66, that means 63.36 milliliter. So 60, that means if you have fermentation, if a fermenter has this much 100 cubic meter that fermenter harvesting tank, then this much water is to be removed for increasing the citric acid concentration from 22% to 60%. Let me end this lecture. I hope you understand this citric acid fermentation process in details, and I try to discuss this process in details, and also at the end I try to discuss the numerical associated with this problem. Thank you very much.